All right, our objectives today, folks. Our objectives today are number one, you should be able to distinguish between endocytosis and exocytosis. You should be able to distinguish between the two of them. And second, you should be able to make a representative drawing of something that is being undergoing endocytosis and something is undergoing exocytosis. So you should be able to distinguish between the two and to render some drawings of both endo and exocytosis. Now, I looked, and these are not, I could not believe it. Okay, maybe we need to add those, but these are not prefixes in our vocabulary. I looked at the beginning on our vocab pages. We don't have endo or exo on either of those. And those are two pretty important scientific terms that I've seen fairly often. So, what do you think the prefix endo means? E-N-D-O. Okay? If you had to take a shot in the dark, what do you think endo means? Okay. Inside. Excellent. Inside. With that being said, what do you think exo means? Okay. Outside, right? Outside. Have you ever heard of something called an exoskeleton? Okay. An exoskeleton, what would that be? That would literally be a skeleton outside the body. Some bugs. Okay, some bugs have exoskeletons. Okay, what about an endoskeleton? Skeleton inside the body. Where's ours at? Our bones are inside our body, right? So, this would be, we would be an endoskeleton. Now, let's talk about it in terms of what we have here. We know that this endo here means going inside. What is it? What do you think this cyto means? What do you think cyto means? What does that look like? Cytoplasm, right? Endocytosis. Into the cytoplasm, if we break it down. So literally, we could break that down as into the cytoplasm. And what would we say exocytosis is then? Outside of the cytoplasm. So literally break these words down, knowing our vocab, and it will help us with the definitions of endocytosis and exocytosis. All right? So break those down and they'll help you out. Well, what are they? First thing we're going to do is we're just going to have a real quick review of active transport. That's your first flip box. You can write whatever you want. You can paraphrase it. You've already got active transport on the opposite page. Okay? So just so you know, if it were me, I would be writing something like the movement of molecules from a low concentration to a high concentration, but we need to have energy present. We have to have energy to move it against that gradient from low to high. So I would put something like that when I am dealing with my definition for the active transport. Okay? We know it needs energy. We know it goes against the gradient. Now, if we look at the picture, your picture does not look like the one I have right here, does it? On your flip book. It looks more like this one, doesn't it? Okay? Now let's look at this. Right here is our phospholipid bilayer. Here's the head, tail, head. So that's the bilayer of the cell membrane here. So I've got the cell membrane. What do you think this guy is? This purple circle there. What do you think that is? Okay. Well, it's obviously something that's inside. Okay. It's connected within the phospholipid bilayer. What do you think? Huh? Why don't we look at our flip book on the other side? What do you think? Anything? Anything? Okay. On the other side, on 53. Look at that. What do you think? What could those be? You don't? Well, read the tab. Huh? What is the tab for the thing that was in? Huh? A transport protein, right? Okay, so we've got this transport protein in there. So this little sphere here is a transport protein. Obviously, which side? Here's the outside of the cell. Here's the inside of the cell. Which side has the higher concentration of blue boxes? Inside the cell. Now, you can see that by our arrow, we are moving inside the cell with the blue boxes. So we're going from an area of low concentration to an area of high concentration. We are going against the concentration gradient. Therefore, we have active transport and we need ATP. We need energy for that to occur, for that transport protein to shift those against the gradient. Okay? Now, what does that have to do with endocytosis and exocytosis? Well, ladies and gentlemen, both endocytosis and exocytosis are active transport. Okay? They're actively transporting. So that number one tells you what? That number one tells you what about either one of those processes? We need energy. Okay? We need energy no matter which one it is. So, let's look at endocytosis. I'll give you a little second here to write down onto your flip card of endocytosis, and then we'll talk about it. What is endocytosis? It's where part of the cell membrane surrounds materials, forming a vesicle that carries the materials into the cell that can then be released inside the cell. So, a vesicle is formed. Basically, if we have these particles like these little yellow ones there, basically what happens is the cell membrane engulfs or surrounds those little yellow spheres, okay? And as it's surrounding them, it's pulling them into the cell. Why do we have this? Well, some of the molecules are too large to come into the cell. They can't pass through the phospholipid bilayer. So we have to have endocytosis to do so. This could be for engulfing food, for taking in food. As I said before, we need energy, so this is active transport. Okay? So if I look at this, okay, if I look at this and bring it over here, let's see if I can't get those. Here we go. Here's the little balls coming along. Okay? One big ball. It is surrounded and pulled inside of the cell where it can be broken apart and transported. Now, if you look what you have, okay, here's the diagram you have for endocytosis. 1, 2, 3, 4. Now, if you look, let's say this is a food particle. That's too big to pass through the cell membrane. So what happens is, as it gets around the cell membrane, It kind of looks like this cell is trying to swallow that, doesn't it? Okay, it looks like a mouth there. What happens is these edges surround, or what we call engulf, that food cell, and then it closes up here. And what you eventually have is you have a vesicle inside of the cell that has that food in it. Okay? That is endocytosis. Exocytosis is our second one. Exocytosis. So go ahead and copy down what you need to on your flip door, and we'll talk about exocytosis when you get that completed. Exocytosis. Okay? Exocytosis, what is it? Well, it's where part of the cell membrane surrounds the materials in a vesicle and carries them out of the cell to be released. Once again, these molecules that are being carried out of the cell to be released, they're too large to pass through the membrane. So they can't pass through the membrane. We have to have exocytosis. All right? Now, it removes waste and other worn out organelles from the cell. So this is a way that, we say that we could store waste in a vacuole. Maybe the vacuole has a vesicle that carries this stuff out to the cell membrane, and through exocytosis, it leaves the cell. Once again, this whole process requires energy, so it is active transport. Here is the diagram that you have in your flip book. So we can see, here is a vesicle. It is carrying these green dots. Let's say these green dots are waste particles that we are getting rid of. We want to take out of the cell. They're too big to pass through the phospholipid bilayer. So what happens is the vesicle gets over here by the cell membrane, and it starts to become engulfed by the cell membrane, and eventually, we get engulfed. Eventually, there's an opening here where all of the little green dots are exited out of the particular cell. So exocytosis is taking the materials out of the cell. Okay? Now,